This lecture will be simply about the problem of assembling fragments of sequences which we face when we have to do whole genome sequencing. So I'm going to give a brief overview of the biological problem, biochemical problem of whole genome sequencing, and two different approaches, hierarchical sequencing and whole genome shotgun sequencing. But for our purposes, the interesting problem here is the computational problem, which is common in both cases, which is to assemble the sequence from a large number of fragments that have some overlap. So we're going to look at two different approaches, overlap, layout, consensus, and basically the general idea of following sequences in a graph where each node, each vertex of the graph represents something that we read, either a part of a read sequence, for example, and the edges are connections due to the overlap, or the Eulerian path problem, which follows the sequences as edges in the graph, and the nodes represent common suffixes and prefixes. And we will see why this representation, even though less intuitive, is more efficient. So the problem here is that automated sequencing allows us to sequence quickly and relatively inexpensively small fragments of DNA of around 500 bases. However, if we have a whole genome, we have thousands of millions of bases, and we cannot sequence everything in one go. So we can only sequence small, short fragments. One solution to this problem when we want to sequence a whole genome is hierarchical sequencing, where we have this genome we want to sequence. We don't know the order of the bases there, but we can fragment it into large fragments and clone them, for example, in bacterial artificial chromosomes, and generate many copies of these large fragments, and then try to map them with sequence tag sites, so fingerprinting and so on, that allows us to have an idea of where in the genome each of these fragments occurs, because there are some parts of the fragments whose positions we know in the whole genome. And so from this we can create a minimum set of these large fragments that we guarantees that we cover the complete sequence of the genome. And now we need to sequence each of these large fragments. So this is where shotgun sequencing comes into play, where we break down these fragments in a large number of subsequences. They are at random, and we use a very large number because due to their randomness, the need to cover at least the vast majority of the sequence of the fragment forces us to create a large number of fragments. And then we need to assemble them in a way that we know what is the order and how they fit together, so that then we can trace the complete sequences. This is something that reduces the computational cost of this step, because now we are doing this shotgun sequencing and assembling only on each of the fragments, but it requires a lot of lab work to do this initial mapping and to know where in the genome each of these fragments lies. So basically this way we need to do all of this work of creating the physical map and sorting the artificial chromosomes in the whole genome using this information, sequence tag sites, fingerprinting, and so on. And this takes a lot of work. But then the problem of assembling the sequence computationally becomes more feasible, because we are only sequencing each of these fragments. And this was the initial approach, in part because also of computational limitations. So if you have very large sets of sequences to assemble in a very large genome, that could not be solved easily in the computer. Another solution that is more used, an approach that is more used currently, is to do the shotgun sequencing on the whole genome. So basically we fragment everything at random. There are some large fragments for which only the extremities can be sequenced, but can be used to build a sort of scaffold that gives us an idea of how the genome must be followed here, but with lots of gaps because we don't have the complete sequences. But then we also have small fragments, a digestion of the whole genome into a large number of small fragments that can be used to assemble the whole sequence, because now we can sequence all of these fragments completely or almost completely, but also using the information about this scaffold to help sort out the different sequences. So basically the problem that we're going to look here at computationally is this problem of from a large number of small fragments of a long sequence, how can we build or retrieve the whole sequence by ordering these large fragments in the correct order. And this is basically the genome assembly problem from a computational point of view. Note that this field of genome sequences is developing very rapidly. So I'm not going to go into detail about the actual processes in the lab and so on. And these examples are just to illustrate the general idea of the problem. We are going to focus here on this common computational problem, which is to assemble the sequence of the genome from the set of fragments that we have produced in the lab and that we can sequence because they are small fragments. So the problem here is that we have a very large number of small fragments. Note that a whole genome can have tens or hundreds of millions of base pairs, and the fragments that we can sequence are around hundreds of base pairs. In addition to that, since the fragments are random, we need a very large redundancy to be sure we don't have holes in our sequence. So to give you an idea, if you want around 99% coverage, that is only 1% of the sequence is not covered by the fragments, the random fragments generated, we need about 8-fold redundancy. That means that we need to sequence in small fragments around 8 times the complete size of the genome in order that we can guarantee they overlap enough for us to have 99% of the sequence. And now we have to basically find a path through all of these fragments that tells us the right order for all the fragments so that we can assemble the final sequence. And note that if we have millions of fragments, we cannot just explore all combinations. This would explode exponentially and it will be completely impossible to do. So we need some clever way of sorting out all the fragments and obtain the equivalent of this, where we know what is the ordering of all of these fragments, so that then we can trace the complete sequence of the genome. Okay, so let's look at different algorithms for this. One simple approach, a greedy approach, would be to compute alignments between all pairs of fragments that we have so that we find overlaps. This can be done with semi-global alignment, for example, where we check if the end of one fragment is aligned with the beginning of the other. And now, when we look at all these pairs, we pick the ones that have the best overlap. That is, they overlap best in the larger region. We join those two in one sequence, and then we compute the overlaps with all the others, and we keep doing this until we don't have anything left. So it's basically something similar to what we did with multiple sequence alignment, but we are doing this with all the fragments, and we are checking just the overlaps at the extremities and joining small fragments into larger sequences, and keep doing this until everything is aligned. This is not a very good approach, not only because of the large memory requirements, because we need to do all the pairs. So if you have 1 million fragments, you need to do 1 million times 1 million alignments to get all the pair overlaps, but also because it is a bit sensitive to errors in the beginning. If you make a mistake and join the incorrect ones, then this never goes back and is never able to fix that problem. So this is not. There are some software that use this approach, but this is not the best approach, at least consensually nowadays. Another way of doing this is to represent all of our sequences, all the reads that we have in a graph, and then try to find a way through the graph so that we cover all the sequences in the correct order. And this will tell us how to match everything together. So this is the overlap layout consensus approach, where we build a graph where each node is one of the sequences that we read, or a fragment of the sequence. We're going to see this. There are some more sophisticated versions here, but we can think of these as the actual read sequences in the nodes. And we connect them with arcs, with edges, if they have overlaps. So if one, for example, the end of this sequence overlaps with the beginning of that one, then they are connected. And if we have overlaps in other parts of the sequence, we can connect here. But then in this layout stage, we can find these contigs by eliminating redundant connections here. So if we have this order of these fragments here, we don't need to care about this overlap here because it's already implicit in this sequence. And then there is a problem here that sometimes there are branches, because you can have repetitions in the genome, and so you can have overlaps that are not really corresponding to the actual sequence of those fragments, but it's just an accident of having some repetitions, some repeated sequences in the genomes. So there are some problems here that have to be solved to have the consensus sequence, also because of read errors and things like that. But the general idea is that we are drawing a graph with nodes corresponding to sequences or parts of the fragments that we read, and the connections, the edges on the graph, correspond to overlaps that indicate these may be part of the same stretch of the sequence. So now what we need is to find a path through this graph that covers all of the sequences that we read, and the order of the nodes that we are visiting in that path will give us the complete sequence. So before I go into a bit more detail on this, I just want to give you an idea of how we can create a data structure in Python to represent these graphs. We already did something similar with trees. We used dictionaries with left and right, and each node of the tree was that dictionary when we were doing the UPGMA algorithm for the multiple sequence alignment. But another way that you can use just to represent a graph is to use a single dictionary where each key of the dictionary represents a node. And then you can have, for example, a list with the nodes that are connected to each particular node. If the graph is undirected, you can just put the nodes once. For example, A is connected to B and C. So we have here this connection, A to B and A to C. And in B, you don't need to put the connection to A again because it's already there on A. So in this case, I use this shortcut of putting the connections only to the nodes that are after each node that I'm considering here. If you have a directed graph, then you may want to put all the edges in the correct directions, independent of the sorted, of the ordering. If you have weights for the edges, then you may need to add, for example, use two lists or use tuples here or some other representation. But the general idea is this. You can easily represent a graph in Python using dictionaries by indicating the nodes and the connections between the nodes. So let's go back to the overlap layout consensus. We have a graph like this where each node represents a sequence, a part of a sequence that we read, and these connections represent the overlap. And now what we need to do is to compute a path through the whole graph, or we can even, since we don't know where the thing starts, What we're actually looking for is a cycle, because we want to go back at the same node at the end, so that then we can think of the genome as a circular genome, and so we don't need to know where to start. And usually these bacterial artificial chromosomes are like that. So in general, we are looking at cycles here where we need to visit all the nodes once, and we return to the same node where we started. So this is a Hamiltonian path or Hamiltonian cycle problem, where we have to go through a graph and visit every node once. The problem with this is that this is a very computational demanding problem. This is in this complexity class, NP complete. So basically, if you had an infinite number of agents trying out different solutions, then you could check for each agent quickly if it had an acceptable solution or not. But we don't suspect that there is no polynomial algorithm for this, although that is not proven. And basically, this when we have large graphs, this becomes very difficult to solve, very computationally demanding. So a different approach here is to represent things differently, and the Eulerian path is named after Euler and had this. The origin was in this problem of the 7 bridges of Königsberg, which was the question was if in this city you could go through each bridge exactly once and visit all 7 of them. So was it possible to do this? And Euler argued that it was not possible. Actually, the formal demonstration came a bit later, but the way he tackled this problem was to abstract from the actual bridge problem and represent this abstractly in a graph. So we have each of these land masses here represented as a node, and each of the bridges represented as an arc. And what Euler said is that you can pass through each edge exactly once and cover all edges only if all the nodes have an even number of edges, or only 2 nodes have an odd number of edges. So this would be the start and the finishing node. If all of them have an even number of edges, you can do a cycle and you can end at the initial node where you started from. But this was a necessary and sufficient condition for the problem to be solvable. And this was not the case in the Königsberg bridges problem, because you have several nodes with an odd number of edges. And so there is no way that you can follow this graph so that you follow each edge, that is, you cross each bridge exactly once. It will not be possible. But this is the Eulerian path, whereas the Hamiltonian path is a visit through all the nodes, and this is the problematic problem here. The Eulerian path is a visit through all the edges exactly once. And this is easy to solve with this algorithm. So basically, this is for the Eulerian cycle, but you can use this algorithm to solve the problem easily. You start with any v. If you're going with the path, you can start with a v with odd degree, but typically we're going to use a cycle, and so all of the nodes have an even degree. That is, they have an even number of edges. And basically, we're going to find a path without repeating edges until we stop again at v. And then for any node in this path that still has unused edges, we repeat this process going from that node u and creating a path, any path, that stops again at u. And then we keep doing this until we cover everything. So basically, we are covering all the nodes in this way until we use up all the edges on the graph. And this is guaranteed to work if all the nodes have an even number of edges. This will guarantee to work, and we'll find this Euler cycle or Euler tour, where we go through all the edges exactly once and we end up in the same node where we began. So we can find this using this algorithm. Basically, the idea is that finding a path through all the nodes is different. Finding the path through all the nodes exactly once is difficult, but finding the path through all the edges exactly once is much easier. And this leads us to this Eulerian path assembly, which tries to solve the problem of assembling the genome by following through all the edges, visiting all the edges once, instead of visiting all the nodes once. And the idea is that now we're going to draw a graph where each node represents the overlap between the sequences instead of a fragment of sequence, and the edge represents the sequences that have those possibilities of overlap. So basically, we can use something similar to the De Bruijn graph. This is just a general idea, because in practice there are also problems with the De Bruijn graph, and the actual sequence assembly programs use variants of this graph, not exactly the De Bruijn graph. But the idea is that each node represents a sequence of a fixed number of symbols that is either a suffix or a prefix of some sequence that we read. And we create an edge from node X to node Y if there is a sequence. That edge will represent that sequence that starts with X and ends with Y. Actually, in a De Bruijn graph, it will have to be the first part of the sequence all the way to the end is X, except the last element, and then the end, except the first element, is Y. So there is only a shift of one symbol from X to Y. But we can get the general idea just by thinking of putting an edge between the two connections if we have the prefix at X and the suffix at Y. That is, the sequence starts with X and ends with Y, and that sequence will be the edge in the graph. And now what we need to do is to find an Eulerian tour or Eulerian cycle, or more generally a path if we don't consider the sequence to be circular, but usually we consider the sequence to be circular. And so we need to follow through all the edges exactly once, which computationally is much simpler to do than following the nodes. So this is an illustration of this difference from this paper here, which I recommend that you take a look. Suppose that we have this genome. This is a circular sequence here, for example, a bacterial artificial chromosome, something like that. We digested this at random in different fragments, and we have these fragments here. And now what we can do is we can consider all the short subsequences of all of these fragments, and we can create a graph where each node is a part of a sequence that we read, and the arcs represent connections because of the overlaps or because we are following along the same read that we found. So basically, the arcs here represent connections between parts of the sequences, and these red arcs represent a path that we can find through all the fragments of the sequences. So basically, this would be a Hamiltonian path that visits each node exactly once. It doesn't visit all the edges, because some of these we are going to ignore, but it visits each node exactly once. And since the nodes represent parts of the reads that we have, once we know the exact order of all of these nodes, we can assemble the complete genome that we had there. Now, the problem here is that finding a path through a graph that visits each node once will require us to find the correct path out of many possible paths here, and it's that problem that is NP-complete. So if we have a large genome, this becomes very hard to solve. What we can do instead is we represent as nodes in the graph, a De Bruijn graph, where we are represented prefixes or suffixes. And we represent an edge here where a sequence has this prefix and this suffix. So AAT starts with AA and finishes with AT. So this is one of the sequences that we have after breaking down all of our reads. and this is another one. They are all size 3, so they are all fragments of the different reads, and the edges connect these relations, these prefixes and suffixes. So in this graph, what we need to find is a way of going through all the edges exactly once, and if we do this, we find a cycle that gives us the exact order of these triplets, of these different parts of all the reads that we have. And once we have this exact order, we can assemble the sequence. And the big difference is that finding this path that visits all the edges exactly once is computationally much easier than finding a path that visits each node exactly once and thus needs to ignore some edges and use other edges and so on. So this is much harder to compute. So there are also, of course, lots of problems in addition to the computational problem. There are repeated sequences in the genome, and this can lead to incorrect matches of the fragments. So there is noise in those connections. There are these long and short interspersed nuclear elements that we have in the genome sequence that may cause different stretches of sequences to appear in very so similar stretches of sequences to appear in very different places, and thus create connections that will not be correct if we are following along the genome. And then there are also these kinds of very common sequences like the restriction site from some of these enzymes and transposons and parts of viruses and so on. So there are lots of these elements in the genome sequence that can really confuse the assembly. And then, of course, there is the problem of the errors in sequencing. If 1% of the bases are incorrect, then all of these algorithms become a lot more confusing, a lot more complex, because they need to take into account those errors. And in particular, these De Bruijn graphs used just simply in this way are very sensitive to base call errors, because then you'll have the wrong connections here and everything will go wrong from there. So in practice, there are lots of complexities in these algorithms to take into account all of these problems, but our focus here is basically on this idea of using the graphs, and especially the increase in efficiency when our graph represents in the nodes the overlaps, that is the common prefix and suffix of all of these small fragments of sequences, and the edges represent the actual sequences that we have read. So if we do this, we can use these Eulerian paths or Eulerian cycle algorithms that are much more efficient than the ones to find the Hamiltonian path. So this the technology itself is progressing rapidly. There are many details here that keep changing over time. But the interesting part here for our course is this combinatorial problem and the way it can be solved using the right kind of graphs and an algorithm for going through all the edges in the graph. So to sum up, we saw this problem, assembling all the fragments, and the big difference here is that if we consider the fragments to be the nodes and try to visit all the nodes in the graph, connecting the nodes by the overlaps, this gives us a very costly computational problem to solve. But if we represent the overlaps or the common suffixes and prefixes as the vertices, the nodes on the graph, and the actual reads that we have, the fragments that we read, are the edges, this becomes much more efficient, because finding paths that cover all the edges is much easier than finding paths that cover all the nodes. I recommend that you read this paper. This is a very short paper, about 3 or 4 pages, but that explains why these kinds of graphs are more useful in genome assembly, and you have that diagram also explained there in the paper. So this would be a good way to look at more details and a different explanation of this.